Much, David, and thank you so much for inviting me. I particularly want to compliment the previous speakers on their excellent presentations and hope that I can add to that from a biomedical perspective, particularly neuroscience. So I was asked to speak a little bit today about how a FAIR applies to your, I would say, your average researcher, right? Because the beauty of the FAIR acronym is that nobody wants to be unfair. Everybody wants to be fair. But often the question I get is you hear this machine readability and persistent identifiers and all of this, and how does one really achieve FAIR? And I also usually have to start my talks, and have for many, many years, I'm trying to justify why one needs FAIR, and particularly in the area of data sharing. Why does one need to implement the FAIR data principles? But the funders are actually taking that part of my talk out of my hands, because at least the National Institutes of Health has recently issued their data policy for data management and sharing, and they've actually given a date that they say January 25th, 2023, this new data sharing policy goes into effect. And it basically indicates that most data should be shared. And given that the NIH is concerned with health, we know that there are subject protections. And I know you're aware of that in the sort of geoscience field as well. So there's a saying, as open as possible and as closed as necessary. So FAIR does not mean that your data are open. So most data should be shared. Data should be fair. But there's an interesting little piece in there that's different than almost every other policy I've seen. And that's that all data must be managed. All data must be managed.
Maryann, we can't see your screen if you're already going through your slides.
Oh, that's probably because I forgot to share it.
Oopsie daisy.
That'll do it. Okay, let me do that. Fortunately, you don't have to see very much. Okay, so here we go. Share. Okay, so basically, as I was mentioning, that all data needs to be shared, or not all data needs to be shared, but all data needs to be managed. And I don't need to show you that slide again, but essentially as a follow-up to that and in preparation for the implementation of this mandate in January 2023, the National Academies of Science, Engineering, and Medicine held a workshop in April 28th, 29th, which I was fortunate enough to chair. And the name of this workshop, the title was Changing the Culture of Data Management and Sharing, a workshop. And it really went through all the things that would be necessary for the NIH to implement this policy, and the research community, the societies, all of the stakeholders into it to be able to implement this policy. But it was interesting that the title really was Changing the Culture. Right? This wasn't about simply a mandate. And the tenor of the conversation throughout these 2 days was really not about compliance checking, right? That you had to check a box that said, I put my data into a public repository and therefore I'm done. The tenor of the conversation was really around this idea of changing the culture. There was a desire of everybody who participated in this workshop, researchers and funders alike, that we want to move beyond this idea of simple compliance, and that data sharing should be impactful and a benefit to all stakeholders. And those stakeholders, again, include both the consumers of data, obviously, but also the producers of data. The other theme that kept emerging over and over again is that stakeholders should be actively participating in defining data management and sharing, that they didn't wish the research community to be passive recipients of this policy, but they, along with their societies and various organizations and institutions who will be at the forefront of this, would actively work to say, what is meaningful and impactful data sharing mean to us? So these two themes kept coming up over and over again, and I thought it was worthwhile sharing that with you, and I'll touch on these again. So the idea that it's more than just compliance checking, but that it should be a benefit, came up in the idea of what is the benefit of data sharing for me, your average researcher. And again and again and again, it said that data management itself is the main benefit. We are all producing large amounts of data. So many laboratories lose control of their data very early, and it migrates as graduate students go out of the lab or postdocs go out of the lab. People lose control of their data. And so again, I'll keep mentioning these themes. The theme of this was that data management itself is a gateway to data sharing, because if one manages one's data properly, then it is not only good for science at large, for all the reasons that you probably heard about this morning, but it's good for you as a person who needs to do research. For example, in biomedicine, underpowered studies are a major problem. So you need to be able to use public data in order to augment what you're doing. As I mentioned, data sharing and good data management are closely aligned, and it helps you with the compliance with all of these mandates that are coming about, both from journals and funders, because well-managed data means it's much easier to generate reports or change your data or whatever it is you need to do in response to a review. On the right, though, we talk about the ones who are going to benefit the most for the longest period of time. And number one is future you. So you as a researcher, how many times have you gone back and looked at your lab notebook and go, I don't exactly know what that means? As I like to say, no one ever regretted annotating or documenting their data too much, but they often regret not annotating or documenting enough. So the one that's most likely to benefit from good data management and sharing is you, because you're much more likely to be able to find your data and reuse it. But also your colleagues. That is, we spend a lot of time needing to share data with our colleagues, whether formally through large projects or else just because a colleague contacted you and said, hey, can I use your data? An inability to be able to get, document, and communicate about that data is a major impediment to collaboration. So if you have well annotated data and well managed data, it's a lot easier to engage with colleagues. And again, if you're a PI, what happens when the graduate student or postdoc leaves the lab? It's much easier for you to be able to recover all the intellectual investment that you've made in that individual without, as one person said, having your right arm cut off every time they leave the lab. So the benefits of data management accrue now, but they also doubly accrue, I would say, in the future, because this data becomes available to you. So there's been now some formal study on data management research practices in the lab, and John Borghi and Anna Gulick gave several talks at this National Academy of Science symposium, where they shared with us what their findings were, because they've done a lot of it. And they have this nice research data management guide inside of their paper, where they basically say, what is the difference between poor practices and optimized practices for reuse? So I'm not going to go over this whole table, but you can see here that an ad hoc practice when planning your project is, when it comes to my data, I have a way of doing things, but no standard or documented plans. I don't follow a consistent approach for keeping my data organized, so it often takes time to find things. And I kind of decide what data are going to be important while I'm working on it, and typically save it onto my laptop or some single location. So those practices actually characterize the majority of laboratories, I would say, in science, and even sometimes my own laboratory, right? So we know that this is not something, as we've been hearing in the earlier talks, that people often pay a lot of attention to. What is the difference if one implements data management practices that are optimized for reuse? So one says things like, I have created plans for managing my data that are designed to streamline its future use by myself and others, right? Always keep future you in this. I organize my data so that others can navigate, understand, and use it without me being present. And I save my data in a manner and location designed to maximize opportunities for reuse by myself and others. And so when one starts to think about data sharing, in the National Academy workshop, there were several speakers who said, you know what, if you want to understand how to share data effectively, stop thinking about what some random person out there whom you've never met is going to need in order to understand your data, and start thinking about future you and your groups of colleagues. What do they need to be able to understand and reuse your data? And although that won't solve all problems, and as you've been hearing all along, we can't solve all these problems now, it will go a long way towards being able to share with the wider community, because at least if you can do that effectively, right, then the steps taken to perhaps add a little bit more metadata for some random user will be a lot lower than if you've taken no thought at all. So the question is, if assuming one buys the argument, right, that you are likely to benefit from good data management practices by yourself, your lab, and other small groups of colleagues, the question then becomes, how do I do this, right? What is it that I need to do in order to be able to share effectively? And believe it or not, even though oftentimes when people start going through the FAIR data principles, one's eyes glaze over because you're like, I don't know, I don't get this, I don't understand what all these things are and why should I care about them? The FAIR guiding principles actually sort of lay out a series of best practices that if you follow them, make it much more likely that you'll be able to share with your lab members and your colleagues. So what are those? The things that I want to emphasize, we've heard the term machine readability a lot during these talks, and machine readability is at the essence of FAIR. Those of us who have been in this field for a long time through the Neuroscience Information Framework and others where we have to deal with public data know that machine readability of data is often not achieved. We've had people share JPEG files of spreadsheets, for example, not understanding that that's not actionable data. So there's been a lot of emphasis in FAIR on machine readability. But I don't want to minimize the human readability too and what that can get us. Humans have to be able to understand it too, not just machines. It's a partnership. It goes hand in hand. Okay, and especially in these early days where acquiring and documenting metadata is difficult and therefore you never quite get enough metadata, for example, using simple conventions like we heard before of file names that are human understandable and machine processable often go a long way. So I think you've seen these before, right? The FAIR data principles aren't just the F, A, I, and R, but there are a set of recommendations that are embedded in there for what it is that you're supposed to do in order to achieve this. And we've heard a lot of these mentioned as we've gone along. I'm not going to go over all of these, but I'll go through a few of them. But I also think it's helpful, especially in the area of data management, to sort of encapsulate what is the high level thing that you're trying to achieve if you want to make your data fair. And so FAIR didn't rise in a vacuum, right? FAIR arose from all these people representing different projects, individuals who have said, these are the things you need to do if you really want to reuse data. So I like to use this nutshell because over on the right here, you have what they call a FAIR data object. And a FAIR data object comprises the data. It's got identifiers, and you heard a lot about identifiers and how core they are. It adheres to some standard. It provides code and other things that you need to understand it. And then above all, it is accompanied by an abundance of metadata. So if we try to sort of distill those down, what is it that we're really asking of people in the lab? What is it that you need to do in order to make your data fair? Well, I thought that these, which came from an article by White et al., were pretty good. Well-documented data are easier to find, understand, and use. As I mentioned, nobody ever regrets documenting their data too much, but undocumented data, very hard for you even after a year to understand what it is that you've done. Properly formatted data, and we heard about this before, are easier to use in a variety of software. So proprietary formats, poorly formatted data, poorly formatted spreadsheets just make it that much harder for you to do anything with that data later on. So one wants to pay attention to that. Data that are linked to other research objects. And again, we heard about this before, like related data, code, publications, even samples, protocols are much easier to use because it provides you all of the context that you need in order to be able to understand this data. And data that are shared in established trustworthy repositories are easier to find and use. If you manage your data on your own hard drive, that is a very bad way these days to be able to manage your data because you're not responsible for data stewardship or other things and all the things that repositories do, as we heard earlier, to make it easier for you to find your data, not just in the week or months after you produced it, but years after you produced it. Those are really professional responsibilities, and institutions and others maintain these types of data services and facilities maintain these types of services so that you will have access to your data whenever and wherever you need it. The second thing I want to emphasize about FAIR, and I think you heard a lot about this in the previous talks in particular, is that you're not really in it alone. As a researcher, there are certain things that you must do as the producer of the data, but you don't have to do everything. As you heard, once you transfer stewardship to the repository, they do a lot of the services for you. So I'm actually not going to talk a whole lot about persistent identifiers, but If you need a DOI for your data, that's not something you do yourself. You do that by transferring your data over to a trusted steward who is going to assign that. It's going to make sure that your metadata are properly formatted and adhere to a community standard like DataCite. We heard all of these sorts of things. So you don't really have to do everything yourself. But the pieces that you do need to do, which is good data management, making sure you have metadata, making sure you have permissions to share, making sure you've documented, paying attention to your data formats, paying attention to the standards that are in use so that you have everything you need in order to be able to submit your data to a repository. Those are things that you do need to pay attention to. So I like to make sure that when I talk about FAIR, I'm absolutely clear that there's certain things that uniquely you as a researcher have to do, but you do not have to do everything. There are other partners in this, particularly the trusted repositories and registries. I also want to talk about community organizations because we heard a lot about that in Kristen's talk. And in neuroscience, we have the International Neuroinformatics Coordinating Facility. And these are convening groups and others that really try to help coordinate across the FAIR landscape, making sure that all of the pieces talk to each other and that things are coordinated. In the particular, INCF, and I'll talk about this later, has been reviewing, endorsing, and organizing community standards, which are so critical to FAIR. And I'll talk a little bit about that later. So these community organizations are also really important partners in FAIR. And I think emphasizing how things cross over and what each organization does and how they can help each other really helps to demystify and decomplicate FAIR, because there's only certain things that you are responsible for. So now I want to go through a few of the FAIR data principles in just a little bit more detail. First of all, we see things all the time that data are described with rich metadata, and metadata and data are richly described with a plurality of accurate and relevant attributes. So a question that researchers often have is, well, what are these, right? What is rich metadata and what is a plurality of accurate and relevant attributes? Data are hungry beasts and they have almost an infinite amount of metadata that one could acquire and present as part of it. And that's often daunting for domain scientists. And I like to say, using that quote that about pornography, right? I'm not going to define it, but I know it when I see it. And you as the domain experts will also know it when you see it. When do you have enough metadata to get started? And when are you kind of lost? So I usually show these examples right here, right? This is an exhibit, exhibit A, I call it, from a repository that was pretty early on where they take MRI data. And you can see here that it tells you some things about what it is, but it doesn't really tell you very much. And you can kind of guess some things, and other things you're like, maybe I don't really know. And once again, I want to say, if you can share with yourself and your colleagues, you will be much better positioned to share with the community at large. So what other types of things would you like to know about this? Well, we heard about some of them before. This is instead OpenNeuro, which is a more recent repository for magnetic resonance imaging. And here you can see you have this nice descriptive title. You know who uploaded it, you know where they are. It gives you information about the data themselves. It gives you README files. It describes the data. It tells you what files are in there. So in fact, if one thinks about this, right, there's a lot of things that if you just did those things, giving it meaningful titles, study purposes, talking about the techniques, listing your authors, all of these things go a long way towards helping others use your data. And is it ever completely enough? No. But at least if there are humans in the loop, you also know the people involved. And for at least the immediate future, you can contact them and ask them for help. And that's often a very big way that people end up reusing data, is they make use of the corresponding author for these data in order to be able to ask questions, okay? So there's all kinds of things that one can do. And I think you appreciate that landing on a page like this or seeing this page in an electronic lab notebook is a lot more informative than seeing this, right? You have a much higher chance of being able to use that data than you do in the first case. So we've already heard a little bit also about provenance as well. And provenance is another type of metadata that is really, really important. And FAIR also emphasizes these qualified references, that is links to other metadata. So you really want to know, if you see a data set, who produced the data and where was it produced? Why was it produced? We talked about some of those in the previous one. You also kind of want to know what produced it, what software tools you, for example, what code did you use, and how was it produced? Perhaps a scientific protocol. And I want to just mention that at least in biomedicine, we're getting a lot of uptake of a tool called protocols.io, which is a trusted repository for scientific protocols. They're given DOIs, they're managed as fair research objects. They make it easy to really share your detailed protocols. And because they have these DOIs, they're very easy to link to your data set. So it says, here's the data set and here's the protocol by which I acquired these. So there are better tools that are coming about for you, not just having to write reams and reams and reams of things in README files, but where you can actually produce some of the provenance and documentation you need using these types of well-maintained tools. Finally, I want to talk about domain relevant community standards. And this was again mentioned over and over again in the previous talks. In neuroimaging in particular, there is a standard that has really started to take hold called BIDS, called the Brain Imaging Data Standard. This was one of the first standards that was endorsed through the INCF process. And I want to say that the INCF endorsement process was really designed to represent the end user, in this case, the researcher, rather than the tool providers or the repositories. Too often, these things get done from their perspective and not what does the researcher need in order to manage their own data. So the standard has to be very well documented and has to present evidence that it actually can be used by the target audience beyond the group that developed it. So I want to spend a little bit on BIDS because I think it's really an example of a successful standard, at least in the field of neuroscience. So BIDS at its heart, and we talked about this a little bit earlier, is a file organization and naming scheme. It takes files from MRI, and as you'll find out if you go and look at the site, there's about 20 other data types that it starts to handle that are related to this, right? They start to create extensions for these specialized ones. So it gives you a way of organizing your data and naming your data. Those of you who are in magnetic resonance imaging may recognize the DICOM data set structure. This is a standard too, but I as a human looking at this can make no sense whatsoever out of what I'm seeing here. In contrast, if you look at a BIDS data set, there are things that you as a human understand. At the same time, it implements regular structures, regular naming conventions so that computers can also parse it. So you could look at this and say, oh, gee, this is subject one, and they have anatomical, functional, and diffusion weighted imaging associated with it. So it's very, very, very simple. And a lot of people in the beginning are like, you know, this is nothing special. But I think one of the important things about BIDS is that it was designed for and with significant input from the researchers who were expected to use it. This was something that they could use in the laboratory. And it benefits from additional tooling, but it doesn't completely require it. And I think that that's really important. These standards have to be usable by the people who need to use them. So what are the benefits of using a standard? Well, it's easier for another researcher to work on your data, right? We heard about that. If a graduate student or postdoc has already left the lab and all you had was the BIDS data structure, you would still understand a whole lot about that data and how it was organized, and you would be able to use it. There's now an entire ecosystem around BIDS that is creating processing pipelines and data analysis software so that there's a benefit to you because you can actually use it in your research. Databases for sharing, such as Open Neuro and a lot of the other ones that are being developed inside of biomedicine, all take BIDS. And that means that if you've used that, you've gone a long way towards being able to share your data. And there's validation tools because it's a regular structure that can help you early on to say, am I doing this right? And it often points out things like, oh, wow, this file is missing. I don't have my metadata. So there's significant benefits that are accruing by using these standards in the lab, provided these standards are usable in the lab. Okay, so I know I'm running out of time, but I just want to again revisit our FAIR in a nutshell. How do I produce FAIR research objects? Well, again, well-documented data are easy to understand. Pay attention to who produces it, what institutions they're at, and what is the purpose. Provide the experimental detail. Provide data dictionaries and README files, right, that say what I did. Properly formatted data are easier to use in a variety of software. If there's a community standard that's well supported, use it because you'll benefit from tools. Think about open formats rather than proprietary ones. And even simple things like file naming conventions can go a long way to help with data management. Data that are linked to other research objects like code, publications, and protocols are easier to use. And as I like to say, Never pass up an opportunity to use a persistent identifier for your samples, for your subjects, whatever it is that makes life a lot easier. And finally, work with your established trustworthy repositories, either at your institution or through facilities like MagLab or some of the other community repositories, because those are in the long run going to be much easier to find and use. They manage the PIDs and they take the data stewardship burden away from you so that it can be managed for the future. And finally, if you're welcome to get access to my slides. I just listed a few of the resources that are available if you're in biomedicine to help with data management. Thank you.